Previously, we have discussed about the vector that when a particular molecule is acting as a vector, it must possess one selectable marker, and that selectable marker will select the transformed cell from the non-transformed cell, that is the recombinant cell from a non-recombinant cell. And we also know what is transformed cell. Transformed cell is a cell which has the plasmid, and the plasmid contains the gene of our interest. So let us discuss the mechanism of selection of transformed cell from non-transformed cell. We have started with the isolation of DNA. We have already digested. We have done the digestion of the DNA using restriction endonuclease enzyme. We have amplified the DNA using PCR, polymerase chain reaction. Then we have separated or purified DNA using agarose gel electrophoresis. We have inserted into a vector. Afterwards, we have seen the various techniques of how to insert the target DNA or the foreign DNA into the host cell. Now the next step is to how to select the transformed cell from the non-transformed cell. Very simple. So we will take the example of PBR322, and both the examples, that is antibiotic resistant gene as a selectable marker, and we will also see enzyme producing gene as a selectable marker. So selection of transformed cell, the first method is using antibiotic resistant gene. Antibiotic resistant genes, ampicillin and tetracycline, right? Now that same plasmid PBR322, now we will not draw all the structures of PBR322, we will draw only a few. Let us say this is the site of, we know, BAM H1, and here tetracycline resistant gene was present. And in this segment, we also know ampicillin resistant gene was present. Here ORI and ROP protein was present, right? So we are inserting. Normally, it is preferred to insert the foreign DNA within restriction endonuclease site because it is easier. Because in this segment only, the selectable marker is located. So it will be easier for us to select the transformed cell from the non-transformed one. Add BAM H1. Because we are using this site only, we have to get BAM H1 restriction endonuclease. It will cut the DNA. So our foreign DNA, let us say this is the plasmid, right? Because this site has been cut it down. And now we have to also add the gene of interest. Let us say this is the gene of interest. BAM H1 added, then this gene of interest will be added here. Okay. Let us say that this is the gene of interest. This is the site where we have introduced the gene of interest. Okay, this black one is the gene of interest, and this is the entire plasmid only. Ampicillin region is intact. BAM H1 we have used, and gene of interest. How to ligate? Using ligase enzyme that we already. Right? Now, initially, what we have done is we have used BAM H1 as a restriction endonuclease enzyme because we are cutting in the BAM H1 site. Gene of interest, using ligase we have inserted the gene of interest into the vector, that is PBR322. This is PBR322. Afterwards, what we will do? We will transform it into the host cell. Right? Let us say the host cell is E. coli. Okay, so it will be transformed into E. coli. Okay, somewhere within the genome of the host cell, that is E. coli, PBR322 has been integrated. Now there are three possibilities. The first one, the second one, and the third one. The first possibility is non-transformed E. coli. Right? no plasmid has been received. Okay, so non-transformed E. coli, that means no plasmid. Second possibility is transformed E. coli. With plasmid. Please understand, this is transformed E. coli. E. coli is transformed, that means it has received the plasmid PBR322. Only plasmid, no gene of interest. Third possibility could be transformed E. coli plus plasmid plus gene of interest, and that is basically, generally we call it as recombinant plasmid or recombinant. So this is recombinant one. We can write it here also. This is R PBR322. R, that means it is recombinant PBR322. Why recombinant? Because it has taken the gene of interest along with the plasmid. Now, these are the three cases. Let us now discuss how to select transformed cell from a non-transformed cell. So case 1, 2 and 3. Here ampicillin and here tetracycline. Let us prepare the culture of these three cells differently in the different Petri plates and add ampicillin and tetracycline. First three set ampicillin, second set of tetracycline. Right? Now, normally we know E. coli cell, the bacterium has no resistance against antibiotics, right? So in the first case, it is a non-transformed E. coli, no plasmid. If there is no plasmid, there is no ampicillin resistance, no tetracycline resistance. If there is no resistance, how E. coli will grow? So negative. No growth of the E. coli is observed. Case number 2, transformed cell is there with the plasmid, right? So as it is plasmid has been inserted into the host cell, right? It is a transformed cell with plasmid, and it is an intact plasmid because plasmid has not been opened up. It is an intact, no gene of interest. If there is no gene, plasmid would not have been opened up, right? So ampicillin and tetracycline resistant genes are also present in the plasmid, right? So they will grow. Positive signal. Like third case is very important. It is transformed cell. That means it contains a plasmid, and plasmid also contains the gene of interest. Right? In biology, we cannot have all the time 100% efficient processes. Some cells may take up the recombinant plasmid, some cells may not take. Some cells may take up only plasmid. That's it. Now, the third cell is transformed cell with the plasmid and the gene of interest. Right? Where we have inserted the gene of interest? In the tetracycline region. In the region of Bar H1, tet R is present. Now, what we have done? We have opened up the Bar H1 site. Within the Bar H1 site, tetracycline resistant gene was present. When we open and then we have inserted this desired gene in this segment, then what is going to happen? Initially, the plasmid was resistant to tetracycline. But when we open once, resistant power is lost. That's it. So now tetracycline resistant will not be present. Ampicillin is there because we have not touched up this site. We have only played with this site. That's it. So if it is ampicillin containing medium, the cells will grow. And in case of tetracycline, cells will not grow. And why cells will not grow? Because we have introduced the gene of interest in this segment only. And after inserting the gene of interest, that resistant power is lost. Tetracycline gene become inactive, and this is called as insertional inactivation. That is activation of the selectable marker gene by addition of insert. Insert means the alien DNA or the foreign DNA. Okay. Now let us understand once again with some different aspect. This is the normal growth medium. Normal growth medium, right? Agar-agar medium. No ampicillin, and tetracycline. Ampicillin is also not there, tetracycline also not there, right? So all these will grow. So the colonies that we will observe or that we will obtain will of E. coli colonies, E. coli colony, right? Then we have to transfer these colonies into the medium that contains ampicillin. So nutrient medium. Apart from nutrient medium, now we are adding ampicillin also. Right? In the first case, normal growth media, no ampicillin, no tetracycline, right? All three will grow. So let us make positive sign for case 1, case 2, case 3. No ampicillin, no tetracycline. Each and every colony will grow, right? Now these colonies are transferred through nitrocellulose membrane onto the ampicillin containing medium, right? Ampicillin we have added. Now out of these three, first non-transformed E. coli, no plasmid. If there is no plasmid, no ampicillin containing resistant. So first case, first cell will not grow. Transformed cell with plasmid, only plasmid, intact plasmid. Ampicillin also there, tetracycline also there. It will grow. So let us mark positive, right? Third is transformed cell. It is a recombinant one. Come back to this recombinant plasmid. Tetracycline resistant is lost because of gene of interest. Ampicillin so we have not touched yet. It is still intact. So if there is ampicillin resistant, then bacteria will resistant again ampicillin. It will grow. So positive sign. So here simple meaning is that now we are interested in these two, transformed plasmid and the recombinant plasmid. In ampicillin containing media, which cells will grow? The cells which have the plasmid, intact plasmid, that is a non-recombinant one. We know we are not interested in recombinant also because ampicillin is present. Now again we have to transfer this colonies through a wooden block technique into tetracycline resistant media. So nutrient media plus we are adding tetracycline, TET, right? Now non-transformed E. coli, no plasmid, so that will not grow. So case 1 in tetracycline region, no growth, right? Transformed E. coli, plasmid is there. This is the intact plasmid. Tetracycline resistant gene is there because it is only plasmid without foreign DNA. So plus it will grow. Third case, transformed E. coli plus recombinant one. Tetracycline resistivity is lost because of the addition of gene of interest. So how would any bacteria will survive if the medium contains tetracycline but no more resistant against tetracycline? Why no more resistant against tetracycline? Because gene of interest we have added within the TET-R region only, right? And that is located in the palmatoid. So these cells will not grow, negative. So from here, colonies we will get colonies of only case number 2, transformed E. coli with plasmid. So let us say we are getting these colonies. Right? Now we have to compare these two. How to compare? As based on the location. Circular colonies are there, here also circular colonies are obtained. So these circular colonies are of those for case number 2. I am writing case number 2 because only plus is with case number 2. Right? Here recombinant one are not growing. So these middle section colonies are our recombinant colonies. This is a clear cut indication because in this nutrient media plus ampicillin, this recombinant plasmid can grow because ampicillin is still intact. And in tetracycline, both one are growing. Here only case number 2, that is a non-recombinant one. Here recombinant plus non-recombinant cells will grow. Here only non-recombinant. That this case number 2, only plasmid without any recombinant DNA, right? So colonies which are circular periphery are of non-recombinant one. Here also non-recombinant colonies are peripheral. Central colonies are of recombinant, that is a transformed cell. Collect that colonies, grow in the fresh nutrient media, that is culture it, and produce or isolate the product of interest using the gene of interest. So this is all about selection of a transformed cell using antibiotic resistant gene. In the next part we will discuss selection of transformed cell from non-transformed cell using enzyme producing gene.